Good morning, everyone, and welcome to the Friday Informatics Institute PowerTalks seminar. And I'm very happy to introduce today's speaker. Dr. Chen Huang is an assistant professor of genetics in Division of Genetics Research. He's also a scientist in the Precision Medicine Institute and the O'Neill Comprehensive Cancer Center, as well as our own Informatics Institute. He earned his doctoral degree in cellular and molecular biology at Vanderbilt University. And his research interests are in proteogenomics of cancer biology and treatment innovations, looking at tumor systems biology at the protein level and computational approaches for and data analysis for finding protein markers and treatments. The name of his talk is Proteogenomics Presents New Avenues for Tumor Systems Biology.
All right, thank you, Amy. Thank you for the introduction and also the opportunity for me to introduce our previous work as well as some of the ongoing research. Now I'm establishing my own laboratory. So good morning, everyone. Today I'm going to talk about the cancer proteome genomics, which is a promising and interesting direction to integrate multi-omics data to drive complex disease. So the reason why we think multi-omics, especially when you have the proteomics, could be so important is really because of the very complex tumor abnormalities. So when people think about the aberrations in the tumor genome, there are all kinds of things can go wrong. Copy number variations, structural variants, somatic mutations, these are readily to be translated into the transcriptome level abnormalities. And these will eventually lead to a malignant tumor growth and proliferation program accompanied by a lot of tumor-specific phenotypes. So because the tumor genome abnormality is so complicated, during the past 1 or 2 decades, large cohort studies, for example, TCGA, has been utilizing multi-omics techniques to profile the global tumor biological or molecular phenotype at different omics level. For example, DNA sequence allow you to detect the genetic level change, and RNA-seq allow you to detect the gene expression or other RNA level alterations like alternative splicing. So one sort of knowledge gap left after we have done all these TGA or other cohort-based genomic study is at the protein level. So when you look at the central dogma and think about how things can go wrong or go abnormal in tumor cells, from the RNA to protein or beyond protein, you have the translational regulation, protein stability, and protein modification, and so on. And those are all readily contribute to pathway outcome as well as other tumor phenotype. And not to mention that protein themselves are important biomarkers and druggable targets. So that's part of the reason that during the past 5 or 10 years, people have started to bring in the mass spectrometry-based proteomics and integrate the protein level characterization to the NGS-based genomics. So this new term, proteome genomics, is really to integrate our multi-omics identification and emphasize the understanding of the protein level to drive the biological and clinical insight discovery of the cancer study. So in the modern mass spectrometry, what we are doing is we extract the protein from the whole bulk tumor tissue, and then we use some specific enzyme to digest them into a peptide. And we use, for example, liquid chromatography to separate the peptide. And then we use mass spec to detect those peptides, which will be further assembled into the protein level. So how do we identify the peptides in the high throughput pattern? Is we construct what we call theoretical protein database, and we simulate all the digestion and the mass spectrum generation. And then just like you align your RNA reads into the reference genome, here we align the mass spectrum to our theoretical spectrum to identify what these spectrum refer or represent in terms of the peptide sequence. And this is really an oversimplified schematic diagram, because there are a lot of things need to be customized to really make this work better for tumor biology. For example, we need to probably incorporate the protein or peptide that are only seen in the tumors, instead of only use the canonical protein sequence to really identify tumor-specific protein. And there are a lot of optimizations are going on for this customized proteomics pipeline, which I will also show you some of our thoughts in my later talk. So there are also some of these large cohort proteomics tumor studies has been going on, and probably one of the most influential studies come from the CPTAC, Clinical Proteomic Tumor Analysis, led by the NCI. And during my postdoc, I have been leading the CPTAC proteomic studies for colon, head and neck, and pancreatic cancer. And all of my results presented today will come from these three publications. So just to give you an overall idea how we generate and collect the data, or what our cohort look like. This heat map showing you that in the head and neck cancer, we collect tumors from about 100 head and neck patients, and we used next generation sequencing, just like what TCGA has been doing, to profile the mutation, copy number change, RNA-based gene expression, microRNA DNA methylation. And then we also use the mass spectrometry to profile the proteomics and phosphoproteomics. And this number basically shows you that by this mass spec-based technique, we can identify and quantify about 10,000 proteins, as well as about 100,000 phosphor sites. And we have also well-detailed annotations in terms of the clinical, as well as the patient tracing data. So these provide our platform for us to really looking at not only the genomic abnormalities, but also their outcomes in the protein level. So this histogram shows you in the global level, the protein and RNA correlation is really moderate, while definitely below what people has expected when the proteogenomics studies has been published. So the median correlation coefficient is around 0.5, means that half of them shows relatively low correlation, and some of them even show negative correlation. And if you lay in the pathway of these different, the genes showing different correlations, you can see that some pathways, they are more likely tend to have genes show moderate or lower correlation, indicating that those pathways are more likely to be regulated in the post-RNA level. And this is also something that we have been looking during our study. Okay, so I'm going to organize my presentation into 4 different categories to show you how we integrate proteomic data into the genomics, or integrate the proteomic data into clinical attributes, and to really translate those multi-omics data into clinical and biological insights. So first category is about protein biomarkers, and this is actually very straightforward because we have collect for the solid tumors, the tumor tissue and adjacent normal tissue. So then we can basically do a pairwise comparison for these 2 different types of tissues and identify the proteins that are differentially expressed in tumor versus the adjacent normal. And we are more focused on the proteins that are upregulated in the tumor tissue, because they are more likely to provide us with more clinical insights. For example, we can here in the head and neck cancer cohort, we can annotate those upregulated tumor proteins by whether they are CD antigens, whether they are druggable targets by the current drugs on the market, and whether they are secreted proteins, making them likely to be biomarkers. And I'm just highlight 2 examples. One is a tyrosine kinase, another one is a secreted protein, where you can see in the RNA level, they either show downregulated or they didn't show a significant change in normal compared to adjacent normal, but only with the protein that you see significantly upregulated. So this argues again that at a lot of cases, protein expression cannot be represented, cannot be fully represented by RNA expression. And we did the similar thing for the phosphoproteomic data. So here is showing you that we lay out the differential phospho and the differential protein at the same plot, and we want to focus on the phospho change that with a higher fold change than the protein, so then we can exclude the confounding effect from the protein level change. And for the phosphoproteomics, we are not only interested in what phosphosites are being changed, but we are also interested in what kinase that are associated with those altered phosphosites, because as you know, that kinase are a very large protein category that can be drugged by, for example, small molecules. And indeed, in this head and neck cohort, when we get the differential phosphosites, we can infer the kinase whose activity are increased in this cancer type. Okay, so I think we have made a good start in terms of showing that the proteomic data alone can generate some interesting biomarkers or also some druggable targets. But we really want to push this identification into a higher resolution. For example, when people talk about the protein markers or tumor-specific proteins, they are really mean those are the whole bulk tumor tissue. But because tumor is a complex tissue type, there are multiple non-epithelial cells being infiltrated. And sometimes you really need to have a better idea what proteins or what gene expression are come from tumor epithelial cells. For example, PROTAC is a promising targeting strategy to target some of these tumor driver proteins. And in this case, you really want to have a better understanding that those tumor upregulated proteins, they are really come from the malignant epithelial cells. So then they are the true tumor drivers. And also people has been using spatial proteomics to do this cell type specific protein detection in a high throughput manner. So we have been trying to use data deconvolution, which is to use some mathematical model to dissect in silico of the whole bound tumor tissue expression into cell type specific expression. So these are some of our preliminary findings. Basically, we improved some of the previous algorithms to make them work better at the protein data. And then this is what we call the knowledge-driven validation. Means that we know that these, by knowledge, they are specifically expressed in either stromal or either immune. And our algorithm clearly show that we can achieve these kind of expectations. And with the pathway level, we sort of see similar expected results. And for whatever we have the ground truth from the purified cell group, we can do a more unbiased validation. Okay, so the next category is that by combining proteomics with the previous understanding, especially those from the genomics or transcriptomics, we can really understand better or more accurately of the tumor biology. And I'm going to showcase several examples. So tumor genome is featured with high instability. And for example, in head and neck, which is a tumor type with a lot of copy number change. So you can easily detect the copy number change at the chromosome arm level. For example, 3q, 8q, those are the chromosomes featured by a lot of oncogenes, and they are the ones that are amplified a lot in this cancer type. And you can also do this in the in the focal level, highlighting the tumor drivers by knowledge that their copy number are amplified in this cancer type. But we want to use proteomic data to push the previous findings based on knowledge into a systematic way. So this is what we call to prioritize the copy number drivers using proteomics data. So we reason that the tumor drivers, they should be recurrent and they should be translatable. Recurrent means they should be observed instead of random, but recurrently across different patients. And translatable is that this copy number change need to be refract the alteration in the protein level. And this is how the protein expression can be powerful. So we design a straightforward filtering pipeline, which basically we filter so then we can identify the copy number change. And then these events can translate it into protein level change. By doing this, we narrow down the global copy number alter to about 200 genes in the head and neck cancer. And we use some orthogonal data set to validate our finding. So this is a DMAP from the Broad. Basically, they use the CRISPR or RNAi to knock down individual genes and ask how these genes are necessary for the tumor growth. And you can see that the copy number drivers identified from our pipeline, they show a lower Achilles score. So Achilles score reflecting how the tumor cells, their growth is inhibited by knocking out those genes compared to the genes before the filtering with the proteomics. Our prioritized genes indeed show much more contribution to the tumor growth. And further, we did some pathway analysis to show that not only we can recapitulate some of the previous tumor driver genes or pathways, but also we can identify some of the new pathways that are involved in this cancer type, and their alteration well, they have been altered in the copy number level. Another example is that I want to show you not only the proteomic data can add the insights into the genomic data, but sometimes the proteomic data can even correct the understandings based on the genomics. So in genomics, there is a 2020 rule basically saying that the genes with larger than 20% mutations being truncation mutation, then the 2020 rule will assign the gene as the tumor suppressor gene. It sort of makes sense because people think that truncation mutation is, in a lot of cases, are loss of function. So when you have loss of function, then these genes are likely to be tumor suppressor gene. But in our colon cancer study, we found some case. One of them is SOX9, and it actually fit into this 2020 rule because in this stop gain, which is the truncation, it accounts for 1/3 of all the mutations. But if you ask a biologist, especially a developmental biologist of this gene, they are most likely they are going to argue this gene is a tumor promoting or oncogene based on the biological knowledge. And there is a conflict of the genomic finding to the knowledge. And this is why we bring in the proteomic data. So what we found is that, interestingly, even with the stop gain or truncation mutation, there is actually an increase in terms of the protein expression, which is argued against the tumor suppressor gene of SOX9. And when we look at the protein, when we look at the mutation location of these SOX9, you can see that for these brown ones, most of their location is is outside the functional domain, but before this lysine residue, which means that it's very likely that the stop gain does not affect the core function of SOX9, but it can remove this lysine. And later we combined with the literature saying that this lysine residue is actually a ubiquitination site, which means this stop gain will remove actually this ubiquitination site, thereby stabilizing the SOX9. So this is correcting our understanding of the 2020 rule, means that not all the translocation are necessarily loss of function, and not all the case where you see a lot of translocation, these are tumor suppressor genes. Okay, so going forward, we really want to push our findings into a more systematic manner. So in our previous publication, we have shown people successfully those well-known tumor drivers, TP53, KRAS, P10, etc. But we are still need to demonstrate that omics data can not only provide us with insights to those well-known tumor drivers, but also some of that not that well-known mutations, or sometimes even the rare mutations. So to do this, we can use protein sequence structure and definitely the protein expression and phosphorylation-based machine learning to predict how the mutations affect protein phosphorylation, because phosphorylation is one of the key components involved in the pathway transduction. And also to use network to link mutations to protein interaction and co-expression, because if you think about how protein function in the network or pathway level, the protein-protein interaction and the co-expression is really some important data modality you need to think about. Okay, in the last talk, I'm going to present you how we can combine our omics data and to put them in a very close clinical context. And one example is the targeted therapy for the head and neck cancer. And the take-home message is that previously, based on the genomic evidence, people tend to think that EGFR copy number amplification, which is a common event in this cancer type. And these copy number amplification increase the EGFR receptor expression and drive the EGFR pathway activity and then the tumor growth. And people or physicians have been commonly using the EGFR monoclonal antibody, for example, cetuximab. The EGFR monoclonal antibody, the way it works is to block the ligand, EGFR ligand binding to EGFR. So one caveat is that the response rate is around 30%. So this is relatively low. So with the proteomic data, we really want to gain some insights. Well, our initial plan is to gain some insights why the response rate is so low. So we think there might be some sort of functional redundancy, or we probably didn't use the correct biomarker to screen the patient. So this heat map shows in our head and neck cancer cohort, where the columns represent the patient, and they are ranked by the EGFR copy number. The red ones are those with high copy number amplifications of EGFR. And then you can clearly see that the EGFR copy number amplification can be translated into the increased RNA, protein, overall phospho, as well as some of the tyrosine residue that are known for EGFR function. And this is the correlation coefficient for EGFR copy number to the other molecular phenotypes. Now, when we look at the EGFR activity, so this is inferred EGFR activity. What we did is we took the previously curated EGFR downstream transcriptional target and used their expression to summarize a score to represent EGFR activity, EGFR pathway activity. And we found that the EGFR copy number or EGFR protein phospho increase does not necessarily corresponding to the increased EGFR pathway activity. And these suggest that maybe EGFR copy number or increased EGFR expression is not a good biomarker reflecting the EGFR pathway activity. So we turn to look at the EGFR ligand. So there are 7 EGFR ligands being reported in the literature. So based on gene expression, we found that 4 of them are upregulated. 5 of them are upregulated in the tumors compared to normal. And when we look at these 5 EGFR ligands, indeed, they show a very good correlation to the EGFR pathway. And then we took in our proteomic data. And here, you can see that when our phospho and protein data together in these EGFR downstream pathways, we found that only the phospho level of the EGFR pathway components are correlated to the ligand, to the EGFR ligand, but not to the EGFR receptor. So this fits very well with the other omics, the gene expression-based inference. And this suggests that the EGFR pathway transduction reflected here as the phospho level change is really determined or correlated by the EGFR ligand instead of the EGFR receptor. Now, if you remember, the EGFR monoclonal antibody it binds to the EGFR receptor and prevent the EGFR ligand dependent EGFR pathway activation. So in this case, EGFR ligand should really be used as the biomarker instead of the EGFR receptor itself. And actually, in the publication, we show that there is a EGFR ligand independent pathway that is better associated with EGFR receptor copy number change. So going forward, we want to develop better ways to analyze the phosphoproteomic data. For example, currently, our phosphoproteomics is good at detecting phosphoserine and threonine, but the tyrosine detection remains very poor. But tyrosine is very important for, there are a big pathway, big category of the pathways, the tyrosine receptor-based ones. So how to better impute the missing values for the tyrosine-based phospho characterization is definitely one important direction to go. And also the kinase substrate knowledge is remaining very poor, comparing to, for example, the transcription factor versus the enhancer promoter combination kinase substrate. The database remains much smaller. So we have been thinking to use the cutting edge network-based approach where, the phosphorylation and the substrate and the other, for example, protein expression, protein abnormal level or other modalities can be incorporated as a node, and to use some other graphic neural network-based approach to not only impute the missing values in the network, but also to better interpret the relationship between each other. Okay, the second example that proteogenomics can contribute to the clinical insights is about the tumor immunotherapy. Tumor immunotherapy is a very hot research area nowadays. And people are trying to come up with different tumor immunotherapy strategies to treat different types of tumor. For example, we know that there are a lot of tumor types, what we call the immune hot, means that they have a lot of immune cell infiltration, and the tumor genetic events, for example, somatic mutation, can be translated into the tumor antigens. So in this case, we can design better ways to recognize the tumor antigens, for example, or at least we can identify more specific tumor antigens for those immunotherapeutic targets. And this is where we combine the proteomics to the genomics or transcriptomics understandings of the tumor genome abnormality. So if you remember, the proteomic database construction, the procedure is, the whole idea is we need to include the tumor-specific abnormalities into our theoretical database. So then we can use this database to discover the peptides that with these, for example, somatic mutations or gene fusions. So we have designed a straightforward yet effective pipeline to identify the mutated peptide, which we incorporate the we incorporate the gene expression and the mutation in our customized database, and we have further pipeline components to evaluate the identification of this mutated peptide. When we apply this customized proteogenomic pipeline into our colon cancer, we found that the colon cancer, some of them are what we call microsatellite instability high, or MS high, in which we know that those have a lot of small indels. And indeed, we found a lot of these indels. They can be translated into the peptide product. Although this has not been experimentally validated yet, but based on some orthogonal validation or based on some knowledge-based validation, we show people that this is a highly promising way to go, because potentially these mutation events, if they can be translated into peptides and presented into the tumor surface, there can be customized, for example, TCR-T therapies, where you can engineer the TCR based on these presented peptide. And this, what I have presented is promising, but we definitely have a long distance remain for us to really bring this into the clinical usage. So the reason is because for a lot of tumor types, the tumor antigen is not origin from the mutation. Instead, there are a lot of other tumor genomic abnormalities. For example, alternative splicing, or structural variation, or some of the what we call the non-canonical RF. They may come from, for example, 5-UTR, 3-UTR, those regions that normally are untranslated into, into proteins in a normal cell, but in tumor, for whatever reason, they are being translated. And those tumor-specific events can actually generate actionable tumor antigens, which means we really need to broaden our views of the tumor antigen discovery instead of focusing only on the somatic mutations. So that's why there has been a lot of groups, including ours, try to come up with better ways to build what we call the customized mass spectrometry library to incorporate those genetic abnormalities into the library. For example, I have told you about the somatic mutation calling for the whole exome sequencing. And actually, there are also like alternative splicing or structural variations can be inferred, for example, using chemo-based approach from the RNA-seq. And nowadays, people have also been using, for example, long-read sequencing. This technique is very powerful to refer alternative splicing and structural variation. And also RiboSeq. This technique can directly detect the actively translated regions. For example, for the non-canonical open reading frame, RiboSeq is a very powerful technique to detect those abnormal translational regions in the tumor. And when you have a better customized mass spectrometry library, you have a better opportunity to identify tumor-specific peptide, and then which will allow you to further prioritize what peptides can be bounded by HLA and be presented as tumor antigens. Okay, so how about the immune cold tumors? Those tumor types are usually known to have low immune cell infiltration, but people are still trying to come up with some ways to reactivate the immune infiltration for those tumor types. And we have also showcased an example in the pancreatic cancer. So pancreatic cancer is commonly known as an immune cold cancer type. And the checkpoint inhibitor-based immunotherapy, most of them are not responsive to the checkpoint-based. So in our pancreatic cancer cohort, we use in silico data deconvolution to infer the immune infiltration for this cohort. So each column represents a tumor sample, and each row represents immune infiltration for this cohort. And we found a small cluster where you have these CD4, CD8, those T cells are commonly involved in the cytotoxic effect for tumor. And we found that they are correlated very well for the endothelial cells. So this makes us wonder, whether the endothelial cells are the sort of the limiting factor for the pancreatic cancer to get the cytotoxic immune cells. Or in other words, does the immune cold pancreatic cancer show, because of they have downregulated normal epithelial cells, and for whatever reason, the immune cell cannot infiltrate it into these cold tumors. So we use our proteomic data to look at some key biomarkers or key factors involved in the normal epithelial function. And indeed, we found that for the purple one, which is the ones with that small cluster, high CD8, high CD4, they have higher level of these, what we call the normal endothelial markers compared to the other three group where the tumors are featured with immune cold. And instead, for those immune cold tumors, they are not only lose the normal endothelial markers, but they gain the pathological endothelial markers. So this is an example where only the proteomic data can give you some clear biomarker indication of the pathology of endothelial cells, or we call endothelial cell reprogramming in pancreatic cancers. And I'm going to skip this, but the whole idea is by integrating multi-omics data, especially the proteomic data, we have built a functional association between the endothelial cell reprogramming, which are likely to be driven by the VEGF pathway and hypoxia pathway, and this will likely to inhibit the immune cell infiltration as well as immune cell function. So this make some sense to biologists because if you think about the endothelial cells, the tumor vasculature is the sort of barrier between the blood and the blood cell and the tumor epithelial cell. So these make us hypothesize that the endothelial cells or endothelial related pathways, they might be targeted to improve the immune cell infiltration or maybe the whole tumor microenvironment for the pancreatic cancer. Okay, so the last category is about patient stratification. And this is really new because people have been doing this quite a lot using genomic or transcriptomic data. This is what people call the tumor subtyping in the large cohort tumor study. For example, in the head and neck, we can easily, using the proteomic data, we can easily subdivide the whole cohort into 3 subtypes. And the different subtype, they are likely to show different tumor driver events For example, the first one is what we call the chromosome instability high. And the third one is called the immune, means that these are relatively immune hot tumors. And what we really need to understand is what pathways or what molecular phenotype are enriched for these different subtypes. And in this case, protein expression and protein phosphorylation can provide some unique insights for these different subgroups. But more importantly, I want to emphasize that because many of the drugs on market, they are targeting the proteins, and many biomarkers are also protein-based biomarkers. So instead of only focus on the tumor subtyping, we also try to bring in the clinical knowledge to see whether our protein-based subtyping can provide better clinical insights. So here we enumerate 3 current head and neck treatment strategies: CDK inhibitors, EGFR monoclonal antibody, which is the one I talked about and this is ligand dependent, and also the immunotherapy. So the idea is, if you think about a patient can be treated by some sort of personalized medicine, then you should have biomarkers. You should have targetable pathways reflecting this the reasoning of the drug treatment. So we bring in the omics data. Some of them are the genomic data, could reflect the very early molecular etiology. Some of them reflecting the, for example, this is RB1 phosphorylation, can be by either biomarkers or can be directly related to the targeting of the CDK inhibitors. But the whole idea is by bringing multi-omics, especially the proteomics, we can better identify the biomarkers and the targetable pathways, because proteomic data provide a better view of, for example, pathway cascade or the protein biomarkers. We can gain better confidence when we enumerate the current pathways, current treatment for individual patient group. And when we show the specificity, we indeed find that the biomarkers or the targetable pathways we identified for each of the patient group, they are not only show specificity when we compare them to the patient following this group versus patient not only following this group, but they also show high increase if you compare them to adjacent normal. So going forward, we really want to go to a deeper resolution in terms of this precision oncology or customized tumor patient treatment. So the idea is, although we can subdivide the patient group into individual categories, but also we need to think about in different disease contexts how this subtyping makes sense. For example, for the chemotherapy, we already know by knowledge that there are a lot of pathways in tumor cells can be activated to counteract the chemotoxicity from the drugs. And actually, different patients from our omics data are likely to show different chemo-resistant pathways. For example, some of them may block the transportation of the drug into a cell. Some of them may rescue the DNA damage by the drug. Some of them might inhibit the drug triggered apoptosis to promote cell survival. So what we have been trying to do is to come up with better, approach to dissect the interpatient heterogeneity. For example, we have been trying to use some sort of omics data decomposition to dissect the interpatient difference. So then we can look at for each patient or for very small patient subset or patient groups, what kind of pathways of these can really account for the patient chemo resistance. Okay, so my seminar ends up here, and I want to do some short advertisement for myself. We are a new group, and I'd really like to let you know that we are recruiting graduate students, and we are also looking for collaborations. For example, for experimental biologists, we are not only rely on them to provide the raw data, but also we can contribute to, for example, data mining to generate a testable hypothesis. For the clinical oncologist, the same idea. We hope that our findings now and in the future can contribute to some useful clinical insights. And for computational scientists, especially in the audience from the Informatics Institute, we really want to hear about, we really want to learn and hear about the new mathematical modeling or computational algorithms that can be adapted into our proteogenomics studies. So that's it. And finally, I want to thank my previous lab. I did my postdoc at Baylor College of Medicine. So we have a very collaborative working environment, and which allow me to develop not only my personal computational skills, but broaden my views in the whole tumor biology field. And the studies I just show you the efforts that from not only my own lab, but also a lot of collaborating institutions or universities involving data generation, clinical sample collection, and also, you know, some of the sort of consulting physicians, where we have a lot of useful inputs for our study. Thank you for your attention. And if you have any questions, I'll be happy to answer.
Well, thanks for the great presentation. You know, really happy to have you at UAB and doing this kind of work. I'm glad that we have an affiliation with you. So just kind of wondering, you know, what are some of the collaborations that you might be looking for? Like, are you looking more for data sources, tissue sources, computational, clinical, or that type of collaboration, or all of the above?
Yes. Short answer is all of them. Yeah. We definitely looking forward to collaborate with the local experimental biologist. And yeah, I mean, as a computational biologist, you know, we want to make the best use of our findings and our techniques. And also, you know, so basically, my group focuses on two directions. One is to develop better approach, computational approach for our analysis. So in this case, we really want to bring in the expertise from the Informatics Institute, where we can apply the cutting edge algorithms into our analysis. And we also look for experimental biologists to validate some of our findings if necessary. And in parallel, we look forward to work with experimental biologists to, to make more impactful of the study. And also, we look forward to collaborate with clinicians if there are some opportunities. Our study can be involved in clinical trials or provide some better understandings for the operation in clinical practice. Yes.
Okay, I see Dr. Barnes has a question. He put it in the chat, but he also unmuted himself and turned on his video. So I don't know, do you want me to read it, or you want to read it or say your question?
Yeah, let me say the question. So when you do peptide analysis to analyze proteins, it's rather like a short read at nucleotide level. So you take the short read and infer everything else from it. But I know from the work that I do on proteins and protein chemistry and also metabolomics is that you see many peptides that actually are not part of a protein, but they represent metabolism of the protein. To think that proteins are not metabolized is wrong. And in fact, some of these small peptides that come from proteins are more biologically active than the protein themselves. This is work we've been doing with the Lung Health Center for almost 2 decades now. And it turns out a tripeptide that comes from collagen has a completely different function, and it is the stimulator of lung disease on its own. And so we've work that was done with Imperial College in London found that by knocking out that particular protein, it turned out to be there was an enzyme that hydrolyzed this peptide. By knocking out that gene, now all the animals that had the knocked out gene got lung disease because they now couldn't get rid of this short peptide. So I just wonder how many peptides, I think in the chat I said non-canonical peptides. So we're all restrained by canonical pathways. It doesn't matter whether you're looking at protein chemistry or you're looking at metabolomics. We're stuck with pathways that people have written before. And what we need to do is to get very sophisticated machine learning by combining phenomenology with what we can measure, even if we don't know what it is. So for instance, you might see a peptide, it doesn't map to anything in the database because the database never had it.
Yeah.
And so machine learning really is help us enormously, and I'm sure that's the sort of direction going.
Yeah, absolutely. Yeah, thank you for the question. Yeah, I think this is something a lot of proteomic people or metabolomic people has been pursuing, and we certainly have great interest in dealing with this, especially in cancer, because the whole cancer growth program will cause a lot of abnormalities, well, not only in protein expression, but just as you mentioned, modification. And a lot of them are related to metabolism change. And also a lot of these non-canonical peptides where if you use the canonical protein database, there's no way you can identify this in the mass spec. And so there are several ways people has been trying to do well, but I would say that we are still in a very early stage to completely understand that. For example, for the PTM, the post-translational modification, so there are tons of different formats of the modification. For example, for the glycosylation, one format will give you, I don't know, probably more than hundreds or even thousands of modification forms, and this will, affect your mass spec search. So, well, maybe we can talk about this offline, but the idea is we try to use, for example, the machine learning you just mentioned, or probably some of what we call open search, where we do not give some very stringent when we do the mass spec search. Instead, we do some open search. We give some unknown X or we allow some sort of variation, either for the amino acid or either for the modification. So I believe this kind of algorithm advancement will start to improve the answer to the question. And also, maybe the sample processing can also help because for example, when we start the phosphoproteomics, if you don't perform the phospholipid enrichment, the detection of this can be very low. So I would imagine that if you have some improved sample processing, allow you to enrich for some specific modification, and that will also help. So in terms of the non-canonical peptide, I think it's a very intriguing field because when people think about the tumor-specific events, either antigens or druggable target, this is something where the genomics or transcriptomics can be combined to create some customized database for doing that. But I totally agree, there are a lot of unknown things to explore.
Sure, I look forward to talking to you more.
Yeah, me too.
Any final questions? Well, we are over time, so go ahead. Last question.
No, yeah, so thank you very much. I was interested in the proteomics area, but the challenge I see is the result is mostly time dependent, time matters. And how do you see this as a biomarker? Because it's more a time dependent phenomenon rather than, a marker that can be stable over time.
Yes, I agree. Yes, when people talk about biomarkers, they can easily, the findings sometimes can be misleading because, for example, like you said, the early diagnostic biomarker will be very different from the findings when people claim that this is a tumor or tumor subtype biomarker. And I think currently what the best the proteomics can do is we only can claim that the protein-based biomarker works only for the tumors that are dissected or the tumors are being incorporated in our study. But in terms of, if you go to more early or more detailed disease context, that would be still unrealistic to put that statement. But I know that there are some computational or some experimental ways to improve this biomarker discovery. For example, people try to use some computational approach to construct these pseudo-time-based characterization and to infer some of the early alterations.
All right, thank you. Thank you for the question, and thank you again for the great presentation. I'm really excited to see this kind of translational research going on right at UAB. So I know many of the people in the audience are from UAB. So if you're looking to collaborate, or or you're a mentor, a grad student or potential grad student who would be interested in doing this kind of research, then you can reach out to Dr. Huang, or I can put you in touch with Dr. Huang. Thank you for everybody for attending and for the great questions. Thanks for the great presentation, and have a great weekend. Hope to see you at the next Power Talk. Thanks very much.
Bye-bye.
Bye-bye.